Okay, so I'm back, and we'll go through some of this. All right. Okay, so oxygen demand. We've been talking about kind of the wastewater context here a bit. And really, oxygen demand is one of the key contaminants that most obviously affects our aquatic systems downstream. So when we're talking about BOD, you guys have probably covered it in other classes before. So we'll kind of go a little quicker through here, but I just want to have these slides in place, walk us through it real quick. Ultimately, what we want to do is be able to model the actual oxygen content in a river downstream. That's kind of what we're going to move towards here. So now that we've treated the water, part of our regulatory framework and part of how we design a treatment process relies on, well, what is happening to the water itself, right? So if we have this biodegradable or biological oxygen demand being discharged, that's the amount of oxygen demanded, and it's oxygen in milligrams per liter is how we define it. Then the BOD is essentially that food that the wastewater treatment is supposed to remove. And the mechanism here is that respiration, right? Oxygen plus the dissolved organics makes energy. We oxidize. We do this ourselves. Breathe out the CO2. And we're also, the microbes will be making new cells. We also have a concept called theoretical oxygen demand. So that's the theoretical maximum amount of oxygen that a particular molecule can consume. So we can use theoretical oxygen demand to calculate the absolute maximum oxygen that could be consumed by oxidizing some compound. So glucose, for example, we could say we have one C6H12O6 plus some amount of oxygen, and we know we have some amount of CO2 as a reaction product and some amount of water. Our typical oxidation reaction. So if we were to do this oxidation reaction, we can calculate, let's say we're going to have 6 carbons. For the 6 carbons, do oxygens next. So we have 12 here on the left, or excuse me, on the right. We have 6 over here. Well, actually, hydrogens would make more sense. So we have 12 hydrogens. So that means we need 6 waters. So we have 6 times 2 is 12 oxygens here, plus another 6. 18 oxygens on the right. We have 6. So we need another 12. So there we go, right? So that's our theoretical oxygen demand. For every 1 mole of glucose, we use 6 moles of O2, right? So then we could convert that to mass and all that. So theoretically, if we knew that all of our waste bacteria food stuff is specifically glucose, we can calculate that. In reality, we almost, well, first of all, we never have that specific information about our wastewater. Almost never. Maybe there's a sugarcane plant that can get close. But also, the microbes are not 100% efficient, right? There's going to be some compounds that are not fully oxidized. Maybe they only oxidize 5 out of the 6 carbons on average or something. So you never quite know, like the theoretical can be useful, can give us some understanding of the maximum oxygen that could be demanded, but it's very, it's like that upper limit, okay? BOD is sort of the practical occurrence, and the theoretical is the upper limit. Somewhere in between is the chemical oxygen demand, and this is sort of I guess, an empirical way to try to measure the theoretical in some sense. So what we're going to do with the chemical oxygen demand is we're going to take a chemical process that creates lots and lots of oxidation potential. We add oxygen, maybe we add ozone, we blast it with high temperature, maybe using UV. Perhaps we use some sort of a catalyst, and we just try to oxidize as much as we possibly can whatever's in the water. So we take a sample of water and we treat it in that manner, and essentially we're just trying to burn it. And we're going to measure how much CO2 comes off, and we're going to use that as a determination for how much oxidation occurred. So, the same type of reaction there. If we had some nitrogen in the organic matter, which is pretty typical, we'd have N2 gas coming off as well. So ultimately, that gives us a chemical method that's pretty quick. We can do the chemical method in probably the course of an hour or so. Some instrument can do that probably with an auto sampler. So it's just a pretty quick method to check how much oxidizable stuff is in there. And that gives us an estimation. So if we were to rate the theoretical oxygen demand versus chemical versus biological, it's going to go in that order in terms of how much oxygen we will ultimately estimate can be consumed. The theoretical is going to be that max. The chemical is going to be next. That chemical method is pretty harsh. Depends on what setup we use, but it's going to be pretty harsh. This is also essentially what we do when we measure what's called TOC. So this is essentially a TOC measurement, total organic carbon. They're very similar. You maybe tweak your analysis to look at how much carbon was there instead of how much oxygen and so things like that. Maybe need to purge your CO2 first. Very similar, possibly even you could use the same instrument for either. Kind of side note there. So biological oxygen demand then is going to be a slightly lesser result here, and it takes longer. So our typical BOD measurement takes about 5 days. It's a little bit arbitrary, but we want to make sure we have plenty of time to get a good measure, a good estimate of how much biological activity is going to actually happen. These microbes are also going to not just produce CO2 and water, they're also going to be making new cells. They're also going to be releasing some stable byproducts in doing that. So they might take organic phosphorus and turn it into phosphate, for example. So releasing some nutrients. Of course, they'll be using some of that as well, depending on what's limiting. You have bacteria that will take organic forms of nitrogen and ammonia and oxidize that and use that as a bit of an energy source, creating nitrite and nitrate ultimately. So all of these things can and will happen in a BOD test bottle. Okay, there's what we might call a subset of biological oxygen demand. Speaking about these, about nitrate and stuff, we can say we have carbonaceous BOD versus nitrogenous, right? So generated from the carbon consumption or generated from nitrogen consumption. So when we think about how carbon is utilized, we take some organic carbon, so we have some R, meaning some molecule with a carbon in it. We add oxygen, whether that's chemical or biological or whatever. In this case, we're talking about just biological. So in respiration, the microbe is going to ultimately take that, separate that carbon off, and react it with oxygen so that now we have CO2 and just that remainder. Obviously, this is a simplification. They can also do that with ammonia, right? So to take it from some compound, and maybe we have R and an NH2, right? Maybe that's the ammonia amine group on an organic molecule, would go through the same process. But it's going to require oxygen in order to oxidize that to NO2 and then NO3. So, by the way, this is not even stoichiometrically correct, right? This would first go to NO2 requiring O2, and then it would say plus O2, but really 1/2, I guess, to go to nitrate, right? If we were to be a little more precise about it. So ultimately what's going on here is we are taking oxygen in order to oxidize this amine group, whether that's ammonia or whatever. So the reason bacteria will do this is the same reason that they'll do it with CO2. It's an energy source, right? The reaction of oxygen with ammonia is like reacting some reduced form of carbon with oxygen. It releases some energy. Okay, so bacteria will do this as an energy source. And these NO2 and especially NO3 are more stable forms of nitrogen in water, and so they will be converting this organic or ammonia type nitrogen into nitrites and then nitrates. Okay, so that's all kind of from the nitrogen cycle. You could take a look at the various routes. Some microbes will ultimately convert from nitrate to N2 gas. That's a little bit more rare. That would be our denitrifying bacteria here. So they'll take the oxygens back off. You usually have to have a situation where they just really need that oxygen, and they have the right biochemistry to make use of the oxygen that's there, because there's no oxygen around in their local environment. You could take a look at just simply assimilation of nitrates and nitrites and even ammonia back into plants. They can use it. Ammonia can be toxic, so that's usually not at high concentrations, but it should be pretty bioavailable to anything. You could look at ammonification, so nitrogen fixing bacteria, taking nitrogen from the atmosphere, N2, and creating NH4 or NH3. The nitrification, taking those forms into nitrate. All of that in the nitrogen cycle, we can kind of consider that. And for our BOD system, really we're talking about the points where we are consuming oxygen for those components. Okay, so when we're talking about BOD, and maybe you noticed this in our book or problems, we often put a little 5 subscript here. That's referring to a standard convention of using 5 days to test biological activity. So BOD 5, that's how much oxygen was demanded by the water in 5 days. Okay, so that's our assumption here when we say BOD 5. You could do 4 days. Maybe that's sufficient. For whatever reason, the industry, the researchers have generally found 5 to be a good standard. 10 is just pretty long time. 5 is a number of days in a work week. Maybe that's kind of helpful. This is just a common standard, a little bit arbitrary. You do want enough time to make sure that the microbes have time to eat and consume like they would in the environment, right? So we want to make sure there's sufficient time there for biological activity to carry forward. So what you're going to do is you're going to take an airtight bottle. These are often done in these 300 milliliter vessels. You'll see them like this. These little culture vials. If you have this clear type, you're going to want to coat it with some aluminum foil or something to prevent light from getting in. Or you could just use amber vials, because you don't want any photosynthesis happening, right? Some microbes can do photosynthesis. You'll have some algae there. You'll have some cyanobacteria. You really want to prevent that. So you want to avoid any internal creation of oxygen from CO2, right? That would totally contaminate your process of understanding how much of this material can degrade and thereby demand oxygen. In nature, of course, you're going to have photosynthesis competing with the BOD, but at nighttime, that's not true. So usually what we're going to consider is the system on the respiration aspect to understand what could go wrong, what could go bad, right? So that's ultimately the issue. Okay, so then the goal here is to allow natural biological activity for 5 days. measure the oxygen before and after. You could even do it during. And we see here they're using these little oxygen meters to measure the oxygen, dissolved oxygen over time. Usually you'll want to eliminate the headspace so that you don't have that extra oxygen supply going in. So it would just be liquid filled into there. Technically, I guess you could, if you wanted to, you could design it and maybe make use of that. But I think typically it's you have a DO meter and you remove all the headspace. Now, if you have a sample of wastewater, let's say, or particularly dirty lake water that you're studying, I say dirty, but particularly rich in organic matter, you might have a situation where the amount of oxygen that's just naturally dissolved in the water that can saturate at saturation is not enough to supply enough oxygen for the bacteria to do their thing for 5 days, right? You might have way too much waste. And so then what will happen is as you're measuring the oxygen over time, you started at 8 to 10 milligrams per liter, you dropped all the way down to approximately 0. Really, your reading will probably say, oh, it's like 0.5, but the measurement just can't detect lower than that. So that's just what it's giving you. And so at some point, you have to have a hard limit to say, okay, well, we can't go below that. So we have to then dilute. So oftentimes, especially with high strength wastewater, when we do this, we dilute the solution with pure water, typically. Sometimes we have to add a little bacteria into it, but that's in order to have sufficient dissolved oxygen. So we'll say we need more O2 essentially, or really what we're saying is less O2 demand. Because we have that limitation on how much oxygen we start with in the bottle, if let's say we had 300 milligrams per liter dissolved oxygen was the saturation point, we might never need it. In fact, we might never even care about how much BOD we discharge into natural waters. Of course, maybe if the wildlife needed 300, that would be different. Okay. All right, so we can get into the calculations. I'll cover this slide, and then we'll stop here, because I'd like to give us a little more time to flesh out the details. On the simple level, the calculation for a BOD experiment is to use the dissolved oxygen initial. And so I'm going to introduce some terminology here. It's pretty straightforward. DO, dissolved oxygen. We have a subscript I, meaning the initial dissolved oxygen. So when we're doing this 5-day experiment, we're measuring the oxygen demanded after 5 days. That's going to be equal to the dissolved oxygen we started with initially. It's at milligrams per liter of O2 minus what we had at the end. So what we started with minus what we ended with. And then we're going to divide that by the dilution factor if we've diluted it. So if we have no dilution, the dilution factor should just be 1. And we can just simply say, oh, it dropped 5 milligrams per liter of BOD, or of oxygen. That means we have 5 milligrams per liter of biodegradable oxygen-demanding substances. In a way, this is similar, the whole BOD term is similar to how we were describing in hardness, the as CaCO3. We're describing bacteria food as O2, or more specifically as dissolved oxygen, right? Okay, so anyway, this dilution factor P here, that's going to be, in the case of wastewater, the volume of wastewater divided by the total volume in the bottle. So VWW, so for V for the wastewater, divided by V for the wastewater, plus volume of the dilution. So if we consider we have some BOD bottle, some portion of that we fill with wastewater. And then some other portion we add some dilution water. Then that's where we're getting a total which is usually 300 milliliters. And we can say that, well, the total, regardless, is going to be, whatever it actually is, it's going to be that VWW plus VD, right? Those are just volumes. And so, you can kind of get a picture of what we're talking about when we have this dilution factor. So what fraction of the total is actual wastewater, okay? So that gives us a way to say, well, if we measured only 4 milligrams per liter change in oxygen, but only 1% of this whole solution was wastewater, then we could say, well, that wastewater then is 100 times that 4 milligrams per liter drop, essentially. Like that's what it's telling us, because we then divide that by that 0.01, which is multiplying it by 100. So hopefully that makes some sense. We're going to be dealing with volumes here in milliliters pretty much always. It's just kind of going to be our standard with the BOD. Of course, the concentrations are milligrams per liter. So we've got liters there. We shouldn't ever really have to convert between the two, because it's the concentration units versus the volume of the vessel. Okay, so I'm going to leave it there. We'll get into slightly more complicated BOD stuff next time, and start into the modeling of it downstream next time.